Dear guests and lecturers of today, please take your seats in front. We shall start on time as we have live coverage. Se pravi, začeli bomo ob uri, kot je bilo napovedano, ker imamo prenos v živo. Tele ure se držimo, ne? Okay. Ja, menda moram kaj povedati. Dobro, predno uradno začnemo, oziroma tisto, kar se bo videlo potem v samem prenosu, lahko da je tudi prenos se že začel, ampak bom potem vsem uradno najavo naredil. Dogodek bo potekal mešano. So this event shall have a bilingual structure, a part of it in Slovenian, a part of it in English. Predvidevam, da večina gostov slovenskih razume tudi angleščino, pa vendarle gre za dogodek, ki je uraden, zelo pomembno gojiti, seveda tudi naš slovenski jezik, nekako pa poskrbeti tudi za vključenost vseh. Prav, nismo na trdno razdelili, kdaj bo v angleščini, kdaj bo v slovenščini. V samem uvodnem pozdravu, kjer imamo več slovenskih gostov, bo verjetno prevladovala slovenščina, potem v drugem delu bolj angleščina, kar pa ne pomeni, da morda ne bom kje pri kom naredil kakšnega čist kratkega povzetka, ne zato, da bi izpadel pameten, ampak preprosto zato, da bodo tudi gostje naši angleški imeli občutek, o čem je bila beseda. Tako da, če bi rad še kdo kaj vprašal, zdaj le še čas, čez pol minute pa ne bo več. Najbrž ne. Moja ura že kaže, da lahko začnemo, tako da čez nekaj sekund pravzaprav bi potem res začeli. Okay. Hvala lepa. Moje ime je Gregor Cuzak, danes sem vaš moderator in tukaj smo na dogodku, ki označuje pričetek delovanja Centra tehnologij celičnih in genskih terapij, pomembnega mejnika v razvoju ne samo Kemijskega inštituta, ampak slovenske znanosti. Še večji pomen pa ima sam Center za razvoj medicine in dobrobiti pacientov. Pravzaprav v samem štartu delovanja, v samem pričetku prizadevanja za to, da pride do tega začetka tega projekta, je bila vodilna misel, predvidevam, tista, kar nas povezuje, recimo nas, jaz delam tudi kot zastopnik pacientov in marsikdo tukaj le še v občinstvu in tudi vseh raziskovalcev in ljudi, ki delate v farmaciji ali pa v zdravstvu, v medicini, ste zdravniki, to, kako pravzaprav koristiti pacientom v tej novi dobi, ki je pred nami in to dobi, ki nam odpira številne nove možnosti. So, dear ladies and gentlemen, my name is Gregor Cuzak. I am a patient representative. There are a few of us who are that, but I think everybody here is gathered on the basis of actually providing benefits to the patients in this new era of science. This is an important milestone for the Chemical Institute, but not just for Chemical Institute, also for the wider community, for Slovenia, for research institutions, and not just Slovenia, but also Europe as a wider whole, including the partners within the project who are the building blocks of this project. So without further ado, I would like to invite the first welcome speech from Professor Dr. Gregor Anderluh, the Director of the National Institute of Chemistry.
It will not be a speech, really, and it will be in Slovenian. Drage dame in gospodje, spoštovani cenjeni gosti, predstavniki ministrstva, predstavniki akademskih inštitutov, predstavniki farmacevtske industrije. Izjemno smo veseli in ponosni, da lahko danes štartamo na tak način ta izjemen projekt, ki smo ga uspeli z našimi raziskovalci tu na Kemijskem inštitutu in s pomočjo zunanjih sodelavcev dobiti v okviru evropskega financiranja. Kot je bilo že omenjeno, to je izjemen korak, ne samo za Kemijski inštitut, ampak tudi širše in to bi vam radi danes tukaj predstavili. Zelo lepo vas pozdravljam, upam, da bodo poudarki, ki bodo danes v tem prvem dnevu te otvoritve in začetka projekta bili koristni. Slišali bomo veliko nekih poudarkov o tem, kako je to organizirano v drugih državah in veseli me, da s tem pomembnim korakom bomo zdaj eno tako zelo napredno infrastrukturo tudi pri nas lahko vzpostavili en tak center. S tem centrom se Kemijski inštitut premika iz tistih osnovnih raziskav bliže uporabi. Tudi to je en od pomembnih korakov našega inštituta in zato je v naši strategiji to res zelo pomemben korak naprej. Ne morem mimo tega, da se ne bi zahvalil vsem vpletenim kot direktor inštituta, v prvi vrsti predvsem profesorju Jerali in njegovi ekipi, ki je poskrbela za to izjemno prijavo, ki smo jo na inštitutu pripravili s pomočjo projektne pisarne, ki jo je vodila takrat dr. Barbara Tišler, s pomočjo kolegic in kolegov tudi na Ministrstvu za visoko šolstvo, izobraževanje in šport še takrat, gospod Tanjo Vertel bi izpostavil tukaj, ki je zelo veliko pomagala pri prijavi. Sedaj pa seveda v naslednjem koraku pa tudi dr. Mojci Benčina, ki je pa stopila zdaj v tiste velike čevlje, ja, in bo zdaj ves projekt vodila in koordinirala. Tako da s temi seveda tudi ta širša skupnost, ki nas je podprla, zdravnike tukaj iz Ljubljane, skupnosti pacientov, mednarodne partnerje, to je res velik projekt, ki ga sami ne bi zmogli, in tukaj res to nek tak, zdaj se ekosistem izoblikuje okoli tega in to upamo, da se bo tudi v prihodnje nadaljevalo na podoben način. Uživajte v današnjem dnevu, uživajte v predstavitvah in upam, da bo to seveda čez 5 let, ko bo nova stavba, ko bodo novi projekti, ko bo že kakšen rezultat, da se bomo tudi v podobnem miljeju še srečali. Hvala lepa.
Thank you to Professor Anderluch. I would like to just emphasize one word that he used in his speech, which is ecosystem. He touched on it at the very end. A colleague of mine told me last week that business schools now teach a subject called leadership of ecosystems, which is not a top-down leadership, but it's a leadership within equal partners, especially if patients are included. This requires a lot of patience with patients. So please be patient with us. And again, without further ado, I would like to invite dr. Valentina Prevolnik-Rupel, državna sekretarka. Najavljam dr. Valentino Prevolnik-Rupel, državno sekretarko na Ministrstvu za zdravje. Valentina, prosim.
Ja, najlepša hvala vsem za besedo, hvala za vabilo. Spoštovani, dear ladies and gentlemen, I'll talk in Slovene as well. Dovolite mi, da najprej izrazim res iskrene čestitke k temu izjemnemu uspehu cele ekipe Kemijskega inštituta za pridobitev tega res velikega projekta iz programa Obzorje. Gre seveda za pomemben razvojni projekt, ki je plod znanstvene odličnosti in inovacij ter sodelovanja med številnimi strokovnjaki. Tudi sama sem šele pred mesecem in pol prestopila iz raziskovalnih vod, delala sem na Inštitutu za ekonomska raziskovanja v državno upravo, tako da vem, kaj pomeni projekt iz programa Obzorje pridobiti in ga tudi voditi ter koordinirati, tako da vam želim vso srečo, dr. Benčina, to je res veliko delo. Predstavljam si tudi vaše veselje ob današnjem pridobljenem projektu in res iskrene čestitke. V Centru odličnosti za tehnologijo genske in celične terapije bo potekal razvoj najsodobnejših in personaliziranih metod zdravljenja. Ta bo vključeval pripravo proizvodov genske in celične terapije za zdravljenje in njihov prenos v klinične raziskave. Tudi za Ministrstvo za zdravje je to vrsten razvoj ključnega pomena, saj le na tak način lahko oblikujemo modern, na pacienta osredotočen zdravstveni sistem. Če pogledate naše usmeritve zdravstvene politike za leti 2024 in 2025, ki smo jih ravno kar objavili, je to eden izmed naših ključnih usmeritev, se pravi na pacienta osredotočen zdravstveni sistem. Center odličnosti bo med drugim naslovil pomembno področje raka. V Sloveniji živi približno 120.000 oseb, ki so kadarkoli zboleli zaradi ene od oblik rakave bolezni. Rak ni ena sama bolezen, rakov je več kot 100 vrst in čeprav smo pri zdravljenju vedno boljši, je še vedno veliko vrst raka neozdravljivih ali težko ozdravljivih. Izjemen napredek na področju znanosti obeta, da bomo v prihodnjih letih znali pozdraviti vse več težko ozdravljivih rakavih bolezni. Prav napredni pristopi zdravljenja, kot sta genska in celična terapija, so izjemno učinkoviti zato, ker so ciljani. Prilagojeni so posameznim pacientom ali skupinam pacientov. V centru odličnosti je individualni pristop do vsakega pacienta središčna točka pri razvoju novih terapij. V centru odličnosti pa se ne bodo posvetili samo področju raka, ampak tudi drugemu področju, ki bo s pomočjo genske in celične terapije lahko učinkoviteje obravnavano. To je področje redkih bolezni, kjer bo glavna pozornost namenjena tehnologiji za pripravo naprednih zdravil za zdravljenje genskih bolezni, za katere še nimamo učinkovitih zdravil. Namen je seveda premostiti vrzel pri prenosu tehnologij iz predkliničnega testiranja v klinično fazo. Napredne tehnologije zdravljenja bodo v prihodnje lahko zdravile številne bolezni, ki imajo vzrok v genskem zapisu bolnikov. Center odličnosti bo prispeval k več kliničnim raziskavam in bo za slovenske paciente pomenil tudi boljšo dostopnost do najnovejših metod zdravljenja. To bo stičišče vrhunskih domačih in tujih strokovnjakov iz različnih disciplin, pacientov in njihovih organizacij. Torej še enkrat na koncu hvala vsem predanim in odličnim znanstvenikom, strokovnjakom, ki sodelujete v projektu. Prepričana sem, da bo zaradi raziskav v tem centru za številne paciente vzklilo novo upanje za uspešno zdravljenje njihovih težkih bolezni. Čestitke.
So the State Secretary emphasized the role of patients in the center, and this being a center of excellence for cell and gene therapies, it really is on the horizon of the Ministry of Health, which is really good news. What about the horizon of Ministry of Higher Education, Science and Innovation? Vabim na oder dr. Jureta Gašpariča, državnega sekretarja iz Ministrstva za visoko šolstvo, znanost in inovacije. Ne vem, če sem prav povedal naziv ministrstva.
Prav ste povedali.
Hvala
lepa. Dear Director, ladies and gentlemen, I have my speech, my address prepared in English, but I shall follow your example and your two and say only a few words in Slovenian. But you may use it then afterwards. Drage kolegice, kolegi, ekipa Kemijskega inštituta, Gregor Roman, res sem v imenu ministrstva zelo vesel in ponosen, da sem danes tukaj. To je že, ali pa rekli šele drugi tovrsten teaming projekt, ki ga v tej državi imamo in je res odraz izvrstnega sodelovanja in pa predvsem predanosti vseh, ki so bili v ta projekt vključeni. Prvi vrsti seveda bi podaril inštitut, drugi vrsti moram podariti moje kolegice z ministrstva, takrat še MIZŠ, zdaj z Ministrstva za visoko šolstvo, znanost in inovacije, ki so ves čas stale ob strani temu projektu. Zdaj, načeloma stališča ministrstva velikih pomislekov pri tem projektu ni bilo. Financer, vse tisti, ki delno plača račun, vedno ima pomisleke pri vsakem projektu, naj se že kako dobro sliši, ampak prav velikih pomislekov pri tem projektu ni bilo. Iz več razlogov. Najprej zaradi tega, ker je napisano tukaj, to mi je zelo všeč tukaj od spode kot moto tega projekta. Most med biomedicinskimi raziskavami in njihovem prenosu do pacientov. Spravi zaradi tega, ker je zelo v ospredju transfer of knowledge. V drugi strani tudi zaradi tega, ker gre za teaming inštrument, ki ga lahko s komplementarnimi viri financiranja izvrstno izkoristimo za to, da uresničujemo to, kar smo zapisali v vse naše strategije, v strategijo pametne specializacije, v strategijo raziskovalno-inovacijsko strategijo, v nacionalni načrt razvoja raziskovalnih infrastruktur. Se pravi, z združenimi močmi, z evropskimi in nacionalnimi viri lahko le dosežemo take prebojne rezultate. Država bo tukaj zagotavljala izgradnjo in nakup visokotehnološke specializirane opreme, ostale stroške pa obzorje Evropa. Tako da še enkrat res smo veseli in ponosni in komaj čakamo, da bo projekt ugledal sadove, predvsem pa še enkrat in upam, da ne prevečkrat čestitke in predvsem uspešno vsem sodelujočim. Hvala.
I will use the same trick as before. I pick a word and then I try to build on it. The word was bridge. All right? So the bridge between the research and application, some call it bench to bed. So it's really super important for us, patients, that what is discovered in the vials, on the bench, in the research really comes to the actual lives of patients, to the bed, where it's treated via doctors, and of course the whole approval process, clinical trials and everything. I'm sure it's a delight and pleasure, as expressed by the State Secretary, to support such excellent projects as this one looks at this moment, but it shall prove its validity in the next years. Zdaj bi pa k besedi povabil doktorja Sama Zvera, vodjo kliničnega oddelka za hematologijo v kliničnem centru Ljubljana.
Hvala. So, license to Slovin.
Prosim.
Okay. Dobar dan. Tukaj sem že kar nekajkrat, tako da se počutim domače, tudi hlajenje ste zrihtali. Mi je pa vedno zadovoljstvo sam priti, ker vidim zelo veliko ljudi, ki jih res spoštujem, ki so mnogo pametnejši kot jaz, ki jim je znanost bila položena v zibko. Meni jaz, kar se znanosti tiče, bolj tako, ampak kar pa znam, je pa na preprost način to videti, kam to paše, tako delo. In kolega Sever, ki je vse to, kar jaz nisem, tako da se nekaj dopolnjujeva, in je opozoril to leta 17, 6 let nazaj. Od takrat naprej se trudimo za ta projekt in danes smo tukaj, ker mi si obetamo korist za naše bolnike. In to ni treba biti preveč pameten. Zdravljenja, o katerih govorimo in ki bodo dostopna s tem projektom, ki se začenja, so danes stanejo za posameznika ranga $500 000. Mi imamo bolezni kokrčte in za vsako tako bolezen lahko najdemo 30 do 40 ljudi. Pa si izračunajte 10-krat, 30 do 40-krat $500 000, pa pridemo na 200 milijonov brez večjih težav. Kdo bo to plačal? Ne vem. Zaenkrat nam grejo zdravila super, po zaslugi nekaterih posameznikov. V Sloveniji imamo vse in še več. Ampak temu ne bo tako mogel, temu ne bo šlo tako naprej. In z znanjem, ki ga goji Kemijski inštitut s profesorjem Miralo na čelu, pa Gregorjem, si obetamo, da bomo lahko za ceno enega zdravili 3 bolnike. Naši izračuni kažejo v to smer. Kar pa pomeni, da bomo lahko našim ljudem ponudili zdravljenja, o katerih lahko sanjajo tudi v razvitih državah. Smo blizu začetne realizacije na enem drugem polju. Prihodnost je pa to, kar govorimo danes. In da ne bom predolg, vedno se potem zagovorim, sem daljši kot vsi ostali. Naj povem, da je danes tudi za nas to ogromen dan in da vidimo cilj pred sabo, ki se mu reče ponuditi neko povsem novo kompetentno storitev za naše državljane. In ne samo to, glede na to, kako smo zastavljeni, verjamem, da bomo postali regijski center za to vrstno zdravljenje. In bi se zahvalil vsem kolegom, s katerimi sodelujemo, v prvi vrsti vama, tudi Inštitutu za mikrobiologijo, Medicinski fakulteti, Kliničnemu centru, Ministrstvu za zdravje, kjer imamo zadnje čase podporo in stvari grejo v pravo smer in ne moramo ne uspeti. Hvala.
Lahko še trenutek, eno vprašanje vam bom jaz postavil, pa bom potem v angleščini komentiral. Nazadnje, ko smo se srečali tukaj, ravno v tem prostoru, ste povedali, da za razvoj terapije, na kateri delate, karte, pri kateri bo ta center, kot razumem, tudi sodeloval, da ste pripravljeni iti na kosilo tudi s hudičem. Ali ste bili na tem kosilu s hudičem in kakšen je rezultat tega?
Dobro, to je prispodoba. Je pa res, da ko enkrat stvar je v denarju, jaz sem ponovadi konkreten, ne ovinkarim, stvar je v denarju in ko enkrat toliko denarja noter držaženega pride vse mogoče ljudi naokrog. Meni je v čast in ponos, da nihče v našem delovnem krožku ni vzel centa nikoli za svoj žep. Da imamo čiste roke, stvari so pregledne narejene. In je pa tako, če hočeš napredovati, pa dobiti neke stvari, ki jih rabiš, sem se naučil pogovarjati se tudi z nekom, ki se 5 let prej ne bi pogovarjal nikoli nikdar. Zdaj me ni problem, ker vem, da je v ozadju nek višji cilj. In mislim, da je to tudi prav način, ker ne nazadnje pa lahko nekoga, ki živimo namreč v svetu, kjer je denar glavna paradigma sveta, vladar, vedno bolj, kar ni prav. In pozabljamo na tisto, kar delajo tukaj ljudje, na tisto človeško komponento, ki nam jo dokončno izbrisal ali pa zameglil še COVID na vse to. In dajmo spet postati ljudje. In mislim, da taki projekti ne samo, da nam se strani znanosti odpirajo vrata, ampak nam odpirajo vrata tudi nazaj v eno humano sfero. Ja.
Hvala. To summarize really briefly, Dr. Zver mentioned that the therapies that are to be developed here in today's world cost an immense amount of money, and there is a plethora of those diseases that need to be healed, and this would require an immense amount of money that is not readily available if we project forward. Now, with goodwill and a lot of humanity, transparency, which he has shown in the project that he is leading, I questioned him about whether he would be willing to meet the devil at lunch, which was his metaphor that he used. And he confirmed, and we see that he's progressing wonderfully on the CAR-T therapy. And it's my admiration of many people for what you are doing. Thank you very much also for the answer. Naslednji, če se ne motim, bo posnet, oziroma se javi preko telekonference. Ali je tu?
Kolikor vem, bi mogel priti z avionom, pa lahko da avion zamuja.
Okay.
Ni ga še.
So I shall still ask if Dr. Doru Leonard Irimie. Are you here? I was asking whether you are here to connect via teleconference. So this is very immersive. Very immersive. Nice to meet you. Here is your microphone. Please introduce yourself and step on the floor.
Thank you very much. Thank you very much for the invitation. Compared to the previous speakers, I do not have the luxury of speaking in Slovenia today, so I prepared for an English speech that I will continue like this. I also noticed that we are well ahead of time compared to the agenda, but I will definitely not speak for 35 minutes because I will have a like 35 minutes presentation tomorrow in the morning, when I will speak in depth about project implementation and reporting and expectation on behalf of the European Commission and the Research Executive Agency. I will nevertheless like to point out a very few important things now. So first and foremost, congratulations for the success with launching this project. Also, thank you very much for the project team that was very cooperative and efficient during GAP stage. There has been a number of changes done for a number of reasons. We will develop on this tomorrow during GAP, so I'm glad that we completed this exercise in a very smooth fashion. So also, once again, congratulations for this, because with the move from Horizon 2020 to Horizon Europe, teaming instrument has become more competitive, and this is the third call organized for the teaming instrument, and this was the most competitive by far. So in 2 stages, call 115 proposal received. At stage 1, 32 were invited to submit a full proposal in stage 2, of which 12 have been selected to be funded. 4 additional ones have been included in the reserve list, of which 1 more in the meantime has also been invited to GAP. So there will be 13. Adding 13 to 10 remaining from the first call to 14 from the second call, that makes very selective number of 37 teaming projects running now, launched during the 3 calls. With regard to the teaming instrument, as you will know, widening program has been introduced first in Horizon 2020. That was in basically 8 years ago, has been introduced. And it was supposed to bridge the gap between lower performing and higher performing countries. And as you will know, teaming action is the flagship action of this program. Projects are generally large size. They are in the range of 15 million EU grant, to which at least 15 million complementary funding is expected. Even though it is a widening program, scientific excellence is still main expectation, but I would like to emphasize a few more specific aspects of the characteristic for the teaming projects that have been looked at very closely by the expert evaluators at the evaluation stage, and will be looked at by the expert reviewers at each of the 4 reviews that we will most likely have on site in Ljubljana. First is complementary funding, and I'm looking at the Ministry of Science and Education here, which has written a very detailed and very legally binding commitment letter in the proposal. So this is something that will be checked in detail, and I trust it will be delivered given the positive experience with other teaming projects in the country. The second issue is international recruitment. I have already seen the proposals that we are targeting at least 20 full-time researchers based on international advertising. So this is something that will be looked at as well during the following time. Autonomy, also noticed in the proposal, in the description of action now in the project document, that the intention is to establish independent private entity, as actually done by other running teaming projects of Slovenia. And I think this is also working towards this autonomy, whether in terms of setting research agenda, administration, hiring personnel. This is very much welcome. And last but not least, the sustainability. As you will know, the grant will end in 7 years, and then the centers of excellence are expected to run by themselves, to stay on their own feet. We do not know yet how does this work in practice, because now we are closing basically the teaming project resulting from the first call, called 2016-17. And in this regard, as you will know, there is a teaming club meeting in Isola on the 28th and 29th September. I trust you have already registered this issue of sustainability, how teaming projects, centers of excellence, in fact, will perform once EU grant is closed, will be discussed in depth there. One more thing to mention on feedback to policy. We will require every now and then for you to fill in a number of questionnaires, which will go very often beyond ticking some boxes. This is not just to do item list. It is something that we do use in our feedback to policymaker, which is DG RTD in the European Commission, because as you may have noticed, the widening program and the teaming action has undergone a number of significant changes from the first call until now. And these changes are actually based on this bilateral dialogue between the parties to the grant agreement. So I think this will be it for the moment. I will talk a hell lot more tomorrow. Thanks again for the warm welcome in Ljubljana, warm on all accounts, and looking forward to hearing from the project partners, if I got correctly from the agenda. Thanks again.
I mean, sometimes this space is referred to, at least in my projection, as the Slovenian equivalent of what is in Finland called the Finnish sauna. But it's better now. Professor Zver actually mentioned that it's better, the window is open, so you are also free to take your jacket off. Thank you very much for very kind words. Mislim, da zelo veliko povzemati ne rabim, bom pa vseeno eno anekdoto povedal. V Romuniji, mislim, da je gospod iz Romunije, če se ne motim, pa se opravičujem, če ni, sem kolegico vprašal, kako vidi Slovence, ne, pa je res lepo povedala. Je rekla, vi ste eksperti zaprti v take male škatlice. Skratka, za nas je izziv sodelovanja. Mislim, da je tukaj lepo povedano, da pravzaprav po teh 7 letih mora center najti stik z še različnimi javnostmi, ki bodo omogočile preživetje. Without, again, to wait for the time, because we are half an hour actually too early. We shall continue with the next part, because there should be this natural flow. I would say nothing wrong with actually finishing with the opening remarks early. This leaves us more time for the presentations, perhaps also a little bit of discussion later on. With that, are there any questions, kakšna vprašanja do te točke? Ne? Dobro. So, we shall start with the partners in the project. The first one to present is Professor Dr. Roman Jerala. So he's the head of the Department of Synthetic Biology and Immunology at the National Institute of Chemistry. Professor Jerala, beseda je vaša.
Lepo pozdravljeni, gospod državni sekretar, direktor, spoštovani gostje, dragi prijatelji in kolegi. So I would like to present some events that came to the establishment of this center and what was the motivation for this center. So at the beginning, of course, I have to start with statement that we live in really exciting times, particularly in terms of science and in terms of life sciences, because there have been many, I should use the word revolutionary developments in the last decade or so, that really made the abilities of researchers in life sciences with capabilities that I never dreamed of, and many others even less. The first advance is, of course, the genome sequences, the amount of information that we have available. We know the underlying cause, the mutations of many different diseases, which is making, of course, this much better, so we don't play in the dark, but we really see what is the underlying cause. In addition to those tools that enabled the genome information, we have the tools now to modify this information, to edit the genome, to repair the underlying cause of many diseases with CRISPR and similar tools. Also, in terms of understanding the mechanisms, now structural biology enabled us to have hundreds of thousands of structures, of proteins, which are really the driving workhorses of living systems. But even more, the application of machine learning to protein structure prediction that only a couple of years old really made huge leap forward, because now we have structures or reliable models of hundreds of millions of proteins, even more. Now we have the tools to modify, to design new proteins, those that have not been really found in nature, and that have some very, I would say, exciting potentials for therapies, and that's also one of the fields that we're active. All those tools and combined with many others are then used in synthetic biology, which is also a new discipline, I would say, a couple of decades old, that is enabling to introduce engineering principles into the biological systems. And now the reason for this project is that we can use those tools to translate them into the personalized medicine. One example, or not example, one field is, of course, to treat the underlying causes of disease with gene therapy, gene replacement, genome editing. And the second one is to use cells, engineered cells that act as a living medicine. And those are really more and more applied for therapy, and they are really the target of this call. Pharmaceutical therapy used to be limited to the small molecules, and there was a big revolution of biological drugs, and now the third pillar is being established based on gene and cell therapy that is becoming more and more powerful and targeted for individual patients, and we can address now diseases that have never been possible to be addressed before. So let's look at our expertise at National Institute of Chemistry. So I can say that we really cover the whole range from atoms to the whole to molecules. So we have the expertise and instrumentation as well, from molecular modeling, protein design to structure determination using cryo-electron microscopy. We have a range of biophysical instruments and techniques to characterize individual molecules or interactions of those molecules. We have the tools of molecular and synthetic biology and genetics to modify the code, the genome. We have the tools of cell biology to analyze, manipulate cells, but also deliver different drugs, such as using lipid nanoparticles. And also we have the in-house, in vivo experimental, I would say excellent, small, but quite sophisticated facility, which enables us to test this before going to patients, and to use it on different disease models, and to analyze the effect of those therapies. We can also say that one of the reasons is that we had some recognition for excellence for two advanced ERC grants in this area, and also one so far proof of the concept projects. And we have developed several diverse technologies in synthetic biology that could be potentially translated for clinics. I won't go into details in each of them, but there are different tools that enable modular sort of engineering principle modification, improvement of the efficiency of activation of certain genes, faster response than used to be with the technology SPOC or member. We can achieve a response within minutes rather than hours as transcription regulation. We can regulate cellular properties based on humanized proteins that are regulated by clinically approved drugs, which then avoid the problems of any potential immunogenicity. We also engineered modification improvement of CRISPR technology that enables us now to better, to have higher efficiency of genome disruption or targeted insertion. So in the area, two main areas of our interest, as you heard already, are cancer immunotherapy. So the one where we are collaborating particularly with Professor Zver and Professor Sever at Department of Hematology. We want to implement our tools or new tools into the improvement of cancer cell therapy based on CAR T cells, to improve the efficiency and safety of therapy, to increase the indication for different types of cancer, and to introduce non-viral, more targeted introduction into the cell genome in order to have more efficient types of therapies. In the area of gene therapy, this really started off with engagement with Professor Osredkar, who I'm happy to be here, and Spela, who really encouraged us to delve into this area. So we're using the CTNNB1 syndrome as a case study to develop new types of therapies for this particular, but also for other types of gene therapy. So this is the consortium that has been organized around the CCNNB1 Foundation, aimed to prepare a replacement gene, but also other alternative types of therapies that we're now testing. And also it involves consultations with ENA and manufacturers to prepare and develop drugs, potential drugs for clinical trials. So those are the two main reasons. The third main reason for this project is really that we realize that what is really required is this triangle of collaboration between basic biomedical researchers, between clinicians, and between the interest of patients and patients' organization. Without of having the complete circle, I think it's very difficult to build with. And since we had really based on our long collaboration with clinicians and recently also with patient organizations, we realized that we should make a step forward if we really want to go towards translations to the clinic. And we realized that what's missing is an organization, an institution that could translate those reagents into the clinical trial. So the organization that could prepare clinical grade GMP drugs for clinical trials, because that's one of the most complicated and expensive steps, and that would be impossible to do on our own as we started. So we realized that we should do something about, and rather to wait from the lotto to win, we decided that we should try to find out where are the possible sources, and we identified this teaming action as the only possibility to really get funding for this type of institution. So I'll briefly just review the last part of my presentation, how this started. So it really started basically slightly more than 2 years ago in May 2021, when the concept was really crystallized that we should apply for this project. And then at this step, Professor Jarno Eule, who unfortunately cannot be today with us, then proposed the collaboration with UCL, which is already providing to be extremely fruitful. And then, as we heard before, it was quite competitive. We are very pleased to come over the first phase, and then in the second phase, it's really the key role of Mojca Benčina, who attracted additional partners, and I'm really happy that we have really nice, very nice collaborative partners, and December 22 we were really very ecstatic to get this funding, and I'm really very eager to start this project, and I hope we'll be able to fulfill the promises. It's not an easy task, the real work will start, but with this type of team I'm sure that I'm confident that we'll be successful. I'd like to thank to the people who really contributed to the success of this proposal. So people at National Institute of Chemistry, I couldn't list all of them, but those that are most important, also with support of the leadership from the Institute of Chemistry, the Ministry of Science Education, our clinical partners, and also patient organizations. And I'd like to thank you for your attention.
Dr. Zver, thank you very much. Mislim, da v slovenščino prav zelo veliko ni potrebno prevajati. Okay, there will be a question,
I thought if my name is here, I have the privilege to ask a question.
You do?
Yeah. So really impressive, as you know. I'm really impressed about the technologies you have and the centers you set up. The most tricky part I find always, and we have also a patient representative here, so actually I'm curious about the chats you had, and we have the same, I would say, challenge in the Netherlands. At which stage and to what part involve the patients? Did you have any thoughts about this? Because I mean, I'm hematologist by training, so we have a very vulnerable patient population. They have all different skills. What's your approach?
I mean, patients are involved in many ways. I mean, in the first place, they're the driving force, the motivation to do this. But in just the formal, we'll include the patients in the leadership of this organization. We'll involve patients' organization in some committees. After all, as we heard, the amount of funding to develop each therapy is quite big. So there are big, important decisions, and of course, at this stage. Besides the scientists, where we can tell what's feasible or not, there will be also clinicians that will tell from their view, and also patient organizations what's really the challenges, the goals. They will help us set the goals, because we can tell honestly, I mean, we cannot promise to fill huge number of different diseases, but the goal is really to develop platforms that could be applied not just for a single disease, but the same next time for next disease. It should be much cheaper and much faster.
And maybe to the moderator, and also what not you only ask from the center here, but also from the expert partners.
I have my mic, thank you. Yes, thank you very much, and this question really puts us in front, I would say. So patient organizations in Slovenia have been here since 60s, 70s, 80s. They were never connected into one umbrella association. My interest in patient organizations came from the fact that I was interested in digital health since 2013, and my first invitation to patient organizations to participate in a digital health event, which I organized, was in 2016. I realized how much they are not up to what we expect using, iPhones and internet, etc. Actually, still today, in many extents, when I invite patients to a conference we have next year, next Thursday, many of the patients don't respond to the question, please apply via this Google form. It's an anathema to them. So it's also even a further anathema, so not a topic to discuss the science behind the medicines. But you will always find certain very progressive patients, such as Spela, who was mentioned here, basically creating a genetic medicine for her son, and you will, in the patients, you will always find there is an argument we had before whether patients are always uneducated. Not at all, because all of us are patients at a certain point of time. With the patients, it's very important that you find an organization that is very I would say, pristine, transparent, and driven by the goal, which is not, let's say, money or a position or something like that, but which is driven by a certain need, usually coming from the families, coming from the relatives. So that's an important point. How to distinguish this? So it's important that these patient associations or patient organizations are true to their nature. Let's put it like that. And that comes through trust. You can't fake trust in the long run. You can put it in small bouts, you know, pretend that you're a dolphin and, you know, put on your fins and try to swim, but you're not a dolphin. And that's the point. You have to find the right ones. Professor Zver touched upon that a little bit. We have to find just partners who are willing to participate. In that sense, patient organizations are the best partners you can get. I can vouch for that, I'm sure. Thank you. Please.
I believe that, you know, patients are kind of kings in our kingdom, if we consider also doctors being within. And I believe that they must know about themselves equal like we know about them, because they're the one who will decide whether to follow our instructions or not. And in order to decide, they must know everything. So sometimes it will take you 10 minutes to achieve this level. On another occasion, 3 hours. Doesn't matter. We have to talk, and we have to respect them, and we have, as a doctors, we have to consider the fact that they are those who will decide. And speaking of these therapies, about, let's say, cards are excellent, have excellent learning kind of curves. Let's say put elementary school cards produced by big pharma. Then you have such Miltenis cards. These are kind of gymnasium. What Roman will do is medical faculty cards with academical, pure academical note. So it's an uprising learning curve from big pharma cards to products Chemical Institute will deliver.
Okay, I suggest we continue. Predlagam, da nadaljujemo. Dr. Mojca Benčina, v marsičem že danes omenjena kot ena od glavnih pogonov pravzaprav tega projekta. Veseli me, da lahko predam besedo. Mojca, prosim.
Okay, thank you for the introduction, and thank you for you all coming here and celebrate the start of the very hard task, which is actually ahead of us. So as you're probably aware, and I am aware even more, it will not be easy. There are several reasons why, but nevertheless, we can start with the first part. What is actually the mission of the center? We have to be clear that the mission of the center is to develop gene and cell therapy. That's the mission. I mean, the technologies for that. And that's for targeting neurological disease and immunotherapy of the cancer. But we have to also to prepare these innovative medicines for use in clinics in Slovenia. So that's the mission of the center. But of course, I mean, the goal is to provide Slovenian patients and clinicians with this access. So with the access to modern, effective treatment with the increased their availability. That means they should be cheaper as it is at the moment. So that's the main goal of the center. And of course, that's not only goal of the center. I mean, we can in a line when we are going to that goal at the end, we can also benefit on other parts, and the other parts are to development of new technologies, which will not be only for our Slovenian patients, but it will be available for all the patients in the world. And of course, bringing biomedicine research directly to the patients and training the researchers as well. I mean, that's also an important part, to train our young, new coming researchers to the field of a new era of developing of the drugs, because this is the era which will really expand and require more and more knowledge. So that's also important part. Encouraging the creation of biotech startups. If you will be listening to the talk of our advanced partners, they will really stress that that's something which we need to consider as well, and of course, which we are already doing, bring the researchers, clinicians, and the patients together, so that we really go into the direction of the common needs. So what it will be the organization, because that's important. The founder of the center, it's National Institute of Chemistry, but all will be development and functionalizing the center will be bringing together with our advanced partners, and it needs to be mentioned them several times, because we can't do this development without them, because they have a vast knowledge of their developing of the centers, and they will really try to persuade us that we don't teach, that we don't learn on our mistakes, but that they are actually learning without the mistakes they made before. So they are really crucial for us. And that's University College of London, then the center Charité Hospital from Berlin, University Hospital Berlin, Utrecht University Medical Center, and Technical University of Technology from Dresden. So what are, we've already heard what we have. We've got money for that, so thank you for EU and also for our government. And there are 15 million euros provided by the two agencies, English and EU, and they are mainly funds for the working staff, so the employment, and then the 15 million from the cohesion funds from our government, so to really build up the center and equip the center to really do the work. Where it will be stationed next to the National Institute of Chemistry, and will provide working space, the labs, equipment, and of course the knowledge. So that's something which is important. And probably all are you asking what's the timeline. We already started with the planning and documentation in 2023, so we are doing this now, and the head of the institute can confirm that we are really busy on that. And then if everything goes according to the plan, and we hope that it will go, we will have operational labs in 2027. That's a task we put. At the same time, employment and equipment, we will building up this as it goes with the development. So what we are going to do, the task and the role of the center will be to go on the basic and applied research, so to develop this kind of technologies for rare disease and cancer therapies, to preclinical phase. And for the clinical phase, we will be working with hands in hands with advanced partners and of course with the clinicians, so that the clinician will take this last part together, because there the patients are involved, so we are not really entitled to do that. So for the end, what will be the benefits? Because that's important. The benefits will be scientific, so we'll be learning during this course, and we'll also develop new technologies. Then there is a benefit of health for the patients, so we will be providing new technologies, easier accessible technologies, but there are also economical benefits for not only for Slovenia, but also to the closer region, and social benefits. At the end, we are actually going to a new direction, so with the gene therapies and immunotherapies, so it's important that also the wider audience really understand what we want to achieve, and also in this endeavor also help us. So it's important that we all work together towards the common goal to get to the best, let's say, transfer and translation of knowledge to end products, so to get the patients benefit of that. So with this, I would like to thank you and give the word to the next speaker.
Mojca, hvala. Bi kar nadaljeval. So I would like to invite Dr. Jane Kinghorn, Director of the Translational Research Office at the University College London. If I understand correctly, this is the bridge that we were referring to earlier today, sometimes also referred to as the mechanism that should drive the TRL low digits to the higher digits, towards the TRL 8 and 9 and so on. So please, Dr. Kinghorn, the floor is yours.
Thank you very much. I just wanted to start off by saying how delighted I am to be here, finally, after what seems quite a long time from the initial discussions 2 years ago. And I'd just like to thank Roman and Mojca and the wider team here for your perseverance over those 2 years, and the hard work that you've put in to achieve one of these grants is amazing, and you should be congratulated on the efforts that have been put in. Of course, now the hard work continues. I also wanted to thank you for your patience, for bearing with University College London as a partner, because sometimes it seemed we would never get here because the UK is not necessarily everyone's favourite cup of tea, but I'm delighted that our government pulled through and gave us the funding for this project. So it's a really exciting opportunity, and UCL, I just put this slide up just to say we are a large, comprehensive university, very extensive, over 11,000 staff, 16,000 staff. So of that, over 3,000 are academics in the life medical science area, with over 6,000 postgraduate researchers. So it's a big effort, and we have a lot of academics to work with. And in particular in this space, there's something like over 250 academics working in cell and gene therapies. So we've heard a lot about the ecosystem. We're very fortunate. Our ethos is to put the patient first, and so we've developed long-standing relationships with a number of hospital partners. I think there's about 9 in our wider community, and I've picked out 3 here. Which is Great Ormond Street Hospital. So that's linked to our Institute of Child Health. So we go from the early stages of life right through to UCLH, which is a comprehensive medical hospital that covers all ages and all disease areas. And then Moorfields Eye Hospital, very focused, as it says in the title, on eyes and eye disease. The ecosystem. This is really, really important. The philosophy at UCL about putting the patient first, it really resonated with Roman's vision around what he wanted to achieve here and the setup. But what's really important is really understanding your local and your national ecosystem, from your hospital's clinicians, from who's really investing in you, from funders, equally from the national funders, international funders, charities, also investors like the VC community. Understanding your local environment, that's important for us in London, that we're addressing local needs in our local population of over 6 million patients. But also the links to government and the understanding of policy and what that can really do. You really have to pay attention to that early on. Work together, really understand how they can support your motivation and how you can support theirs. So this is where our philosophy of health and wealth really helps in that. And the UK government has really invested heavily in cell and gene therapies. We have the cell and gene therapy catapult, which is aiding spin-out companies to really address that, one of the first hurdles in manufacturing. They've invested heavily. The MRC and LifeArc have invested heavily in manufacturing capabilities for the academic base. So that sort of influencing that policy is really important. You've done a great job already in terms of convincing the government here to invest, and I think that's a terrific ambition that you've brought into. Industry, really important partners. Translation is, I say it all the time, is a team sport, and no one single organization can achieve what we all want to achieve, which is impact in the society that we live in, impact for our patients. And industry, some academics still view them as a bit of a dirty word, but actually in order to progress products to the market, they are a really valuable partner because they do share their knowledge and know-how and help you overcome those later stages in terms of getting marketing approval through. But first and foremost, patients and public. So I'm really delighted that you've got patient representation right at the heart of the centre, proposed centre, because it's incredible. Unless you understand their particular needs, you're going to be developing things that perhaps aren't so important for the patients to be addressed. So really important to see them at the centre of everything. Okay, this is my last slide. So as I said, UCL is a large organisation with a large number of researchers in this field. The Translation Research Office, I joined UCL in 2009, and we started with three people. We're a large team now of 17 people. And really we're out there trying to identify new projects, talk to the research base to understand their aims and ambitions and exactly how far they have taken their technology, and really help them navigate the complicated ecosystem within UCL, but also make partnerships with external organizations to help them progress their science. So in those first early steps, we've got some mechanisms to really help incentivize academics to work with us and also to identify those first early projects. And we call that, we have some early career research funding, which is about £10,000. That's to aid the early career researchers in getting their first grants and progressing along the translational pathway. And we see this as a talent pipeline. So this is an incentive for us to identify the talent that's out there and help them along that pathway and nurture that. This then feeds into our pilot data scheme, and that's about £75,000. It's about enough money for 1 year's project to take a TRL level 2 stage project to maybe a TRL level 3 or 4. And this is really important funding in order for us to build the product pipeline. And then that allows us to begin to work with those people to help them understand the development path. And then it helps us untap public funding. So the figures here are all for the cell and gene therapy pipeline. So to date, we've managed to help academics raise over £115 million of public funding. 6.7 million of that has come from industry. So at this early stage, industry are interested, but most of it comes from the public funding route. We've then taken those projects. We've supported 73 cell and gene therapy projects in the last 10 to 12 years, and we have 32 projects that are currently active. And it's about 50% in the preclinical stage and 50% of those 32 projects in the clinical phase. We work hand in hand with our tech transfer office. So we do not do IP protection. This is UCL Business's area, but we advise on IP very early on. So we have a whole host of translational education programs here at this early stage so they understand some of the hurdles that they're going to face. We don't want to put them off, so we try and introduce them to that gently. But it's really important that you know where you want to go and some of the hurdles that you're going to face, so you make the right choices early on. So, how's this pipeline developed over the last 14 years? So we currently have 14 spin-outs in the cell and gene therapy area. Some of them are listed here on the right-hand side. We've licensed. You shouldn't forget the opportunity to license your technology to an industry partner. And this is a particular point in case. In 2013, we licensed a technology to BioMarine for haemophilia, and that's recently, last few months, got approval by the FDA. It's called Roctavian. We will begin to see some financial return from that now. But you can see that's taken 10 years of collaborative work with BioMarine to get to that point. So it's not a quick process, but by having support mechanisms and learning from one another, we can help try and speed that up. And that's why we're delighted. We hope to learn from our partners here at the National Institute of Chemistry, our partners in Utrecht and Charity, and we hope to share our knowledge as well. So thank you very much.
I have a question. So the cycle that you have shown us, basically the progression of ideas going onwards to projects, finally becoming spin-offs. How is this in terms of sustainability? Do you see sustainability on the horizon? Where are you in that regard?
So as I said, when you're taking projects from a very early stage, it takes a lengthy time before they get marketing approval and begin to be sold. Yeah. In the deal making that our partners in UCL Business do, they obviously build in stage gates for money to return back to UCL, and there's different ways you can do that, but essentially you can build it as a milestone. The first time you get clinical trial approval, potentially there might be a milestone stage gate there where some money returns. So in terms of UCL, we have invention revenue share agreement. So some goes back to the inventors, some goes back to the inventors' departments, and some goes back to the university as a whole. Right. So there's different models, but it takes time.
Understand. Kakšno vprašanje, some questions? Discussion, please.
I may one.
Yeah, please do so.
So you mentioned in the beginning you're really searching, sort of like looking for truffles, what are the precious things. And I think many of us are researchers and know that also editors for many journals don't realize that we have treasures, yeah, because they reject our paper at whatever level. Yeah. The question is, how do you build up your team that they really are able to detect the treasures?
Yeah, that's so we it's a good question. We have myself and a number of the original team had our training in industry. So I left after doing my PhD and joined pharma. And during that 16-year period, I understood what it was what to look for and questions to consider in terms of what makes a good target, how do you validate that target, how do you progress it through the various stages, preclinical, regulatory, into the clinic, etc. And you get an understanding of those generic questions that you need to address for a development, whether it's a small molecule, an antibody, or a cell and gene therapy. So what we're looking for in a person to come into the team, first and foremost, is an understanding of that kind of applied knowledge and how they would advise academics. Secondly, they have to be able to communicate. It's quite a tricky thing, conveying some alien concepts sometimes to the academics. So communication skills really, really important. sometimes it's hit and miss. Sometimes we don't employ the best people, and they find it difficult to be able to talk to an academic and think, okay, you've got something there. But on the whole, we look for people that can actually converse. So I always start, well, tell me about your research, and then I get them to talk about their research, and then you can pick out some things that we think, well, that's a little bit nearer a translational path. So let's start on that in a little bit more detail. It takes a lot of time. You need to go and meet busy academics according to their timetable. It takes time. And meanwhile, we have projects which they need to serve as well. So it's quite a difficult balancing act. But yeah, it's one we have to tread.
Only a short question for me, it's possible?
I mean, this is the table of professors, so you should be asking. They are just doctors there, so I observed actually the difference. So please, Professor, do.
Yeah. Okay. Also, in line with the question about treasures, mostly it is the situation that we learn during the development phase many uncertainties, to solve or to detect and to solve. And sometimes it is a very risky project we are dealing with, because we cannot expect what will be happen with the patient. We cannot expect the side effects, neither the short nor the long lasting. And how you communicate this with the physicians, and most importantly with the VC, because the most important point is that we see, at least in Europe, is not very willing to go in high-risk projects and have a good feeling for this kind of treasures we at the end earn.
There's a couple of things in there. I'll start with VC investors. Just like companies, if you've dealt with a Novartis and a Pfizer, Alnylam, they are different beasts, all of them. That's the same with VCs. And for VCs, I think it's around, for them, they know what they're looking for and how it's going to fit their portfolio. So when you're communicating a risky project for VC investment, you probably have to go around and, as the phrase goes, kiss a lot of frogs, because you don't really know what the rest of their investment portfolio looks like and what their appetite for risk is. But generally, they're pretty risk averse, in my opinion. At the moment, it's a really bad time for VC investment in this space. It's difficult. In terms of funders, they have different motivations. So in the TRO, when we're looking for gems, we base that on unmet needs. Our tech transfer office, on the other hand, they have to really look with a hard commercial lens. So we might support something and see it through a much more charitable funded route. We also work on a joint portfolio, which is a little bit more commercial. In my experience, I think the waves of risk-taking just change. I mean, all it takes is for one bad example in the press, and then that's the whole, that's the whole field put back a little bit further. But on the other side, we're seeing some amazing advances, and the potential of these is really being pushed by the patients, and that's why they're so important.
We have a question in the audience. Please introduce yourself.
So, GP Shavo from UCL as well. Two comments. They are not a question. The first one is about your question and the fact that it deserves a very long discussion. And I think that we need really to bring to the VCs and the funders the concept of bidirectional research, that it goes so from discovery to patient, but then we needed to take from patient back to the bench. because clearly we have one element of uncertainty that may progress. I'm working in the neurological space, so we are roughly 20 years behind cancer. And I think that we should really look at cancer, how the things evolve with the better discoveries done in cancer, with the toxicity but done in cancer, and so on. But I would like to add to the presentation of Jane by providing an additional bonus that the UCL can provide, which is the diversity. So I think that we in the UK have invested quite a lot in diversity of patient, in diversity of a collection of genetic data. And we can really bring the added bonus of diversity to Slovenia. Because I think that one of the problem of the unplanned discoveries is when we go to the patient, is the diversity of the patient that we treat. And I think that we may have done progress in that direction that we can bring to the consortium and to teaming.
Thank you very much. I suggest we continue with the next speaker. This is Professor Dr. Jürgen Kubal from the University Medical Center in Utrecht. If I need to correct my pronunciation, please do so now.
That's perfect. Thank you so much.
Thank you.
And I'm thrilled for multiple reasons being here. So first of all, when I entered the center, I saw it's an institute of chemistry. And most don't know, my dad is chemist and was working his whole life at a university. So it was a flashback, me waiting in the car for him coming back from work. The second one is access, because you said access is very important. And I'm active since more than 10 years in the EBMT, first in the board. Now I'm chair of the Legal and Regulatory Affairs Committee, where access of advanced therapies is one of our major goals. And when I saw the request of you, to be honest, I quickly checked your science, which was excellent, and the country, and this was a perfect match. So I'm really happy that you invited us. And the third one is a very simple one. When I just had food, I had a Wiener Schnitzel. Yeah. And this reminded me of how close our cultural connection is. Yeah? And I think these are what these grants are about in Europe, in particular in these difficult times, that we remind us where we are coming from and that we connect and try to really work together. So I will talk about the cell and gel therapy programs at the UMC Utrecht. I have a couple of relevant conflicts of interest. You can see also on our webpage, and we will see also later. I have for sure too many slides, but luckily some of the speakers were not too long, so I will steal 5 minutes if I'm allowed. And first I will go on our research focus. And 15 years ago, when I left the United States, I was really thinking what's the best thing I could do as a young postdoc when I went to the Netherlands. And I started just out of the blue because I was impressed by some papers coming out about gamma delta T cells, that I will work on about gamma delta T cells, a cell even most immunologists do not know very well. Luckily for me, this was a good educated guess, because nearly 10 years later there came a paper out where they looked at the clinical outcome of patients and the number of tumor-infiltrating cells, and to the biggest surprise of everybody, not the alpha beta T cells, which were the kickoff for all the checkpoint inhibitor treatment, were the number one, but the gamma delta T cells. And the most frequently studied, and in each conference you go, the NK cells are actually very bad T cells. So, and a lot of other papers came out confirming really the important role of gamma delta T cells in cancer immunotherapy. Doesn't say that the gamma delta T cell is the only one, but really a cell I would like to propagate, and luckily I know in London they have also very prominent gamma delta T cell people who are in the field. The gamma delta T cell even became more prominent in the area of CAR T, because what happened is that people transduced alpha beta T cells, that's what they thought. They had this famous first responding patients, and they looked then 10 years later what are the long-term surviving cells in these patients who apparently might keep in check the leukemia, and by accident, they transduced a gamma delta T cell wearing a car. Wow. Then people said, yeah, but that's an accident. And the London colleagues, I think, had a second cohort where they looked also in their cohort, what is the long-term persisting cell in these patients. And the story is a little bit more complicated because they show a first wave of alpha beta T cells, but also here they found long-term persisting gamma delta T cells, making the point that these cells may be totally underappreciated in a Sleeping Beauty moment, and I think is now really discovered from the community. Within our research team, so it's sort of a deal package you get with me. I just put the 5 prominent researchers in there. The one I'm really happy to join, and maybe Jolt can stand up so that everybody can see Jolt. Yeah, too shy to stand up or wave at everybody. Sebastian, who is one of the person of the first hours, who is working on prediction models. I will show a little bit of this later. We have Trudy Stratemans and Colin De Haar, who is working on ATMP development. We have people working on clinical trials, and we are working also a lot of on bispecific formats, because the ATMPs and bispecifics are, in my perception, also from the clinical perspective, very close, and you need to deal with both. Meaning, we are working on intellectual properties, writing patents about new leads, which is an urgent medical need for solid cancer. We are a lot of thinking about how do we engineer receptors, and I saw already a lot of interesting techniques where we might create unique win-wins with our unique receptors, but also think unique techniques you have. We have really went through extensive immuno-oncology modeling. How can we predict that the one or the other choice is the best choice? Because we know as researchers we have too many choices and what is going to the clinic. And we made really the bridge from having the idea in the lab, making the manufacturing in the lab, bringing it to a full-grade GMP environment as you're planning here. And I can tell you we made many mistakes. Yeah, and I hope that we can tell you a little bit about our mistakes. And I learned also that the London colleagues made similar mistakes. So if mistakes are happening twice, don't do it a third time. It doesn't benefit. And that's why I'm very happy. And I think we had really also good publication output, the fun for the scientists. Some education about gamma delta T cells. If you never heard it, there is one important message, that there are really two main subpopulations. And we'll mainly talk about the delta 2 positive. So if you go into immunology, do research, never look only at gamma delta T cells alone. You need to look at the delta 2 positive and the delta 2 negative. That's really like CD4 and CD8 cells or NK cells and T cells. They have complete different biology. I will focus in this talk only on the delta 2 positive. And the idea is, and we had a couple of papers on this, that you have molecules which are called butyrophorins, BTN2 and BTN3, which are mainly in a healthy state scattered around in the cell. And what is happening if a malignant transformation is happening, then with a small GTPase rho B, there were Scholt had a nice paper out, this is coming to the cell membrane, and it's like you're swimming in the ocean, the shark is biting, the arms come up, and this is seen by the gamma delta T cell receptor. How exactly this looks like was not very well known, and we have been lucky with our models, I will explain later, to be part of a paper which is just out in Nature, where they show that these molecules interact closely and come together, and like this, with a certain transcriptional program, this process is only active in the cancer cell, but not in a healthy cell. So I think this riddle is nearly solved. And I know there's another paper in Nature in Review where they show even the binding crystal structure to even furthermore solve this issue. And this was for us a long time with talking to investors an issue, because people didn't exactly know how this works. We showed here, in this case, in breast cancer organoids, because that are the organoids we are working a lot in Utrecht, that this mode of action can be modulated with certain chemical compounds. And then if you look at different clinical cohorts, an example, low-grade glioma, you see if the gamma delta T cells are there, if the signature is there, then the patients have a better outcome. In line with the first paper I showed, but a little bit more sophisticated, making again the point that gamma delta T cells might be very important at cancer for cancer immunotherapy, and we really need to dive into that. And if you just think about the diversity of these cells, you could put one cell next to the other, go from the Netherlands to Slovenia, around to the Netherlands, Slovenia again, and then maybe to, I don't know, half a globe again. This is the high amount of diversity of these receptors, meaning not everything there will be a germ or a treasure, but there will be, I think, a lot of interesting receptors in there. You need also to understand diversity. It was said UCL understands diversity. I think you need to also understand diversity for gamma delta T cells. That's from a healthy individual. We cloned a lot of different cells. And then from a healthy donor, you see in the blue a very active clone, which is high in frequency. But you see in red one a very low active clone, which is also very high in frequency. Meaning that, and that I think was the downfall of the gamma delta T cell field, that people try to expand from healthy donor gamma delta T cells, infuse them into the patient. They stated it's safe, but it was also non-effective. Yeah, and I do not know any drug which doesn't have a side effect. So meaning you really need to pin down the diversity, the individual receptors, and that's what we do. It's all therefore about, in life, creating a balance between activation and inhibition. So either you would like to have activation in terms of cancer, but you might use these cells also for autoimmune diseases. Then you can more regulate the inhibitory part up. And then also, if you look at all the commercial activities, that's the most recent review in Nature Biotechnology, you see that most of the companies play now with diversity, activation, inhibition of gamma delta T cell receptors. We ourselves started many years ago Gadeta, which has been one of the Nature Biotech spin-outs in 2016 and is still alive. You see a lot of people use also gamma delta as a carrier and then introduce a CAR just to tip the balance in the one or the other direction. The concept we pursued was we wanted to piggyback, and that's also what I learned from our UCL colleagues, on old ideas. Don't make things too fancy. We just took CD4 and CD8 cells, because I'm coming from the TCR and CAR-T engineering field, and introduced there then a gamma delta T cell receptor where we said this is really a highly active one. And like this, we tip the balance, not only in a gamma delta T cell, but really in CD4 and CD8 T cells. We take this innate receptor out of its normal environment. And to simplify this, because usually if I make a scientific talk, then later people say, well, I didn't get it, so I tried in an easy way. Imagine about a gamma delta T cell receptor. It's a very active compound, but it's in a desert. Yeah? And there it just cannot grow. And what we do by transferring it into an alpha beta T cell, which is the last cell living in a patient, you really get a blooming meadow because they are in the right environment. And that's the idea of the whole concept. This took us many, many years. This was when after I came to Utrecht, I think the first key publication on the receptors, on the concept. We then changed the lead because we had a better receptor. We worked until 2023, as you can see, at the mode of action. Still not completely solved, but I think the most important things are solved. Scholz worked a lot on efficacy and safety models. And at the end of the day, what you plan to do here is the ATMP translation, which is a very demanding process. So once you just made a transduction in the lab, that doesn't mean you have a stable process in the clinic. And we started a clinical trial. This took us many, many years because of changes in leads, because of many mistakes we made in the translation. But at the end of the day, we presented now at EBMT some of the first dose escalation data. The first dose level was not very exciting. We saw it's an AML cohort, acute myeloid leukemia cohort, some stable diseases, but this can be a smoldering disease. And in the second cohort, we saw really a convincing complete remission. Somebody 30% blast in the bone marrow, complete pancytopenia, who restored completely with his bone marrow. So very excited about it. Now we entered the dose 3. No toxicities, so let's see how the story goes. But what are the lessons to learn out of this? And I think this is what I propagated everywhere. The TEX 001 story, how we called it, from bench to bedside, needed 15 years. Yeah, and it's just commercially not a viable situation. I mean, for me, it was a perfect career, to be honest. Yeah, but if you think from a commercial perspective, that's really not what you would like to have. And we are still not finished. And what are the models and infrastructures you need to shorten this development cycle of ATMPs? And I think this is what this consortium is about, how we can make such great, crazy ideas sometimes into a shorter cycle that you can have 5 different transduction techniques and make early go and no-go decisions and also quick lead developments. That's why we have been supported and lobbied in the Dutch government a lot. They call them national growth funds. We have been lucky to be part of the first one, which is about regenerative medicine, but has also an ATMP facility. We got a structural grant, which is under the lead in Utrecht from Trudy Stratemans, one of our team members from the KWF, that's the Dutch Cancer Society, where we unite all the ATMP facilities in the Netherlands about standard operating procedures, things nobody would like to share. And I can tell you it's still a very emotional process, so it's not that everybody is sharing now, but we're in the process of sharing. We got this last year another grant which is also about viral production and with a lot of innovation with companies who develop new devices for viral production, which might be also interesting for here. And I think the biggest one we got is Oncode Accelerator, which will be officially launched, so I'm not officially allowed to tell, but I think in 4 or 5 days, which is a 400 million euro program from the Netherlands, where 40 million euros or 60 million euros goes to cell and gene therapy for the development of these infrastructures we need there. So another reason why I thought it's a perfect win-win to team up. And of course, Slovenia was the next one. The other one I cannot announce, but I can tell you that will be also very interesting for here, because this will be, has to do something with artificial intelligence and how to deal with CAR-T. If we go to the next one, so what is then important? So this one meets lead detection machinery. Oh, that's in black, very smart from me. And then the next one is lead prediction machinery. And that's where really Jolt comes in with a paper he made with his colleague Florijn Dekkers, where we looked at breast cancer organoids. And the idea was to take our tech concept and try to understand how these techs work with solid cancer. And then you can do a lot of co-culture preparation, 3D live imaging, image processing, and single-cell transcriptomic. And the smart thing is you can image up to 150,000 cells, and you do multi-omics, if I say also what Scholl says, and then combine this also with the single-cell sequencing data and try to understand who of all these cells you're transfusing are the good cells, which are the bad cells, which cells might be not as good but still very important. And the findings were quite amazing because, like in whole life, you have all states. Some are dying, unfortunately. Some are static. Others are very lazy. Others are slow scanners, high scanners, tickle, they just come to the cell, tickle and go away. And others are really engagers and super engagers. And the question is, which composition of this product you need? So we infuse CAR-T and think that's one product, but that's not true. That's a million diversity of a product. And the product changes also between the patients. And really understanding how this works is important. And just one glimpse how we dived into this, and I hope this works now. So you see in the yellow, it's a breast cancer organoids, 2030 organoid from a patient. The purple one, a bluish one is the tech, the engineered cell. And then if you look at this one cell, it's docking to these whole tumors. And this is about hours. And is then spreading its tentacles around this tumor and is slowly biting into the tumor. And as it slowly gets red, is really eating up the whole tumor mass. And then the understanding should be, what is the property of this cell? And then you can do from the attached cells single cell sequencing, and then you find a lot of new genes, which are in this case about morphological plasticity. You're not surprised if you see this picture, which you really say this is an important property, and that's just the tip of the iceberg we're looking at. Another paper now nearly accepted, hopefully, in Nature Immunology, where we show how we can tip the balance, that it goes to the one or the other right, and I think this is exactly maybe also tools which might be very interesting depending on the choices you are doing here. This is the picture of the different cells in this example. So we have the text, that's the concept I described. We have a WT1 TCR, which is not the London, but the Seattle WT1 TCR, and a CAR-T, and just compared the profiles. And the nutshell is a CAR-T frequently dies, we know this, but is very hyperactive. A TCR is a slow scanner, but also super engager sometimes. And you need to tip the balance somewhere in between. And finding this and having all the choices, therefore I said also the CD19 CAR-T is nice, but that's just the tip of the iceberg. If you would like to be innovative, you need to be somewhere in the middle, and we have a lot of tools to make it in the middle. Potential future here, for example, thinking about CAR-Gs, where gamma deltas are carriers, but I mean, this can go in every direction we are thinking of. The most important one is then ATMP optimization. How do you make your drug? And I think the colleagues from London, ourselves, we have similar approaches where we have biofabrication because it's linked to regenerative medicine, and also the GMP simulation facility under the leadership from Trudy Stratemans, and the GMP production facility under the leadership of Colin Dahar. And the idea, the center part of this is a GMP simulation facility, which hopefully makes it quicker to go from the lab to the clinic. And the question is, what is this? So basically, it's exact same equipment as in the GMP clean room. You have there the interface between researchers and the specialists and other experts, because they're completely different, speaking two different languages. If a researcher thinks I just take another medium because the other one is not available, it's a major change from a GMP perspective. Yeah, and these different worlds need to be connected. And we believe that this efficiently allows the translation from clinic to the clinic. And I heard from the London colleagues, they made the same experience. And you can also do all the documentation at a way much lower price because you do not need to pay the clean room price. There we provide then trained and qualified personnel, and I think that's also an idea that might be interesting to link to the idea here. We have special tech transfer processes. For example, we are participating, maybe you've seen a paper about tumor-infiltrating lymphocytes from our colleagues from John Haanen for melanoma patients, which is getting into ensured treatment for patients where we participate in the production process. We develop a lot of analytical methods and, of course, the GMP-compliant documentation. We have all these folders of documentation you need just to make one product. And then the other people in the clinic asked, why does it take so long? Just because they don't understand. And I think really bringing this understanding to the clinicians, to also your authorities, and I was really thrilled that so many authorities were showing up here, because you really need to have also some stamina to go through, but it's really worthwhile. Because it really makes a change. I mean, in the Netherlands, just as a clinical example, CAR-T was approved by EMA, but not paid by the insurance companies. And it has a 30% survival benefit long-term for patients. So basically, 20 to 30% of Dutch patients died because they didn't have access. And I think, to my best knowledge, the same is happening also here. Because you don't have access, you lose 20-30% of young patients, of young ALL patients, which could be otherwise easily treated. So therefore, I'm even more happy that this is happening. So take-home message is gamma delta T cells are the new black in cancer therapy. That's not from me, that's from this review, but I think it's really interesting. Understanding cellular receptor diversity is key. Targeting innate immune receptors is really interesting, but the challenge is in safety and efficacy testing. So we have to have smart models like mouse models, 3D models. And I think it's worth the challenge. We saw a complete response of an idea I had 15 years ago, just now. But you really need to work on the speed on the technology transfer. Brings me to thanking a whole team. Scholt, who's here, Trudy, Dennis, Monique de Witte is another one, and of course a whole team working on this stuff. Thank you for having us and inviting us.
Thank you very much. I think we've made up on the time that we were a little bit too fast before, but still we have a few more speeches to go. So I would introduce Professor Dr. Petra Reinke. She's coming from Charité, so Berlin. Yes. And please, the floor is yours.
Okay. I will go back a little bit more to the organizational part, what we learned and what we have let's say, established at the Charité to build up an ecosystem of cell and gene therapy. So, and I will start with what was already mentioned and discussed. And this is that we as a clinician have to face, do we have a pointer here? We have to face the situation that in Europe, more than 30% are affected patients affected by chronic diseases. And this is related to the diseases. Some of them are already mentioned. One is cancer, of course, but also other immunological diseases, infection diseases, but also the field of transplantation is an enormous tool. So, and this means that we have now with cell and gene therapy not only an access to help the patient and to enter a global market, even as an academic or clinicians, but it means also that we have tools in the hand which allow us to either in vivo by gene therapy and correction of dedicated, mostly monogenic diseases, or ex vivo to give rise to, let's say, health or to, let's say, overcome the disturbed immune balance, which was already mentioned. But I think one important point we have to realize, and therefore it is so important that we get connected. And this is shown in this cartoon, let's say. First of all, you see that in particular in phase 1 and 2, academic institutions are more prominent in comparison to industries which take then over the lead. And we have another problem, let's say, as an European citizen, and this is that we are strongly behind in more or less all aspects, all phases of cell and gene therapy development. And this is not only the point that we lose the affordability and all the things like that for our patient. Of course, I'm a clinician. I think this is, for me, the major driver. But we lose also knowledge and so in technology development. And by the way, whatever we discuss here, yeah, Jürgen, everything is related to a close connection between biology and technology. Without appropriate technology, we will not have any kind of knowledge, but also no any possibility to translate something to something meaningful, either in the diagnostic or in the therapy. So, and with this in mind, we at the Charité start several years ago to develop, let's say, our translational hub for cell and gene therapy. And based on the expertise we have at the Charité, a little bit different. We focus on products for supporting endogenous regeneration, on biomedical products, also tissue and cell combinations, and of course, gene editing of monogenic diseases and tumor therapies. But one point is quite clear, it was mentioned by Jürgen, manufacturing is a crucial point. How you manufacture and how you characterize the patient on one hand, because mostly we start with autologous products and not with allogenic or IPS-driven or whatever. This is the future, this is what everybody, in particular from the industry, is expecting, to make something off the shelf, yeah? But for regulatory aspects, we have to start with autologous products, which means, per definition, it is a diverse product, because you cannot combine your product coming from you and coming from me, yeah? We are in many aspects different. So, and therefore biomarker, whatever biomarker in general means, but biomarker development and application is one important point. It is not the case that biomarker at any time help us to can be used in a clinic as a stratificator, which is normally, if you look up proposals, what the researchers saying and applying for. It is more a matter to understand the biology from the patient, from the product, and how it works in the patient at the end. And you spoke shortly about regulation and EMA and regulatory aspects. We have several manufacturing authorizations. So for phase 1 to A to B. Products mostly developed by our own. Yeah, so, and I can tell you the first product we developed, this is not only a matter of time, it's also a matter of documentation. And we had for the first product, only one, which is not related to all renewals you have to do, it was 5-kilometer paperwork. And only for getting an approval for the blood, this is the source we are working with, not leukapheresis also, but we focus on blood. It took us 3 years to get the approval, and it took us, this is only 13% of our wound production process, and we end up with a height of documentation like the Eiffelturm. Yeah, so to get an impression, what does it mean at the end to start with a clinical trial? So our government, in particular our Wissenschaftsrat, Council of Research, we can translate this, was fine with our concept. We get 30 million for building up a new GMP facility, which is around 10 clean rooms. And the intention of our center is early phase IITs, speed up, of course, transdisciplinary, this is what also was mentioned, and transdisciplinary, I will come back shortly, to develop innovative technology and in a scalable, cost-effective way, to develop manufacturing standards and cell product analysis, and this means we have to cooperate with everybody. And this is a cartoon showing you what we mean with, let's say, transdisciplinarity. At the end, very slow, we have a discovery team, we have an enabling and technology development team, and we have the clinician. And what we have established is a joint project team dedicated to each product which is of interest for a dedicated indication. And we have defined, which is also not easy, I have to say, different go and no-go checkpoints at different stages towards the clinical trial. And it was also mentioned here the aspect of refined translation, to have this kind of iterative improvement. So because if you come up with a new ATMP, whatever it is, for whatever the indication is, academia take over mostly the whole pipeline, right? And we can do it on an academic GMP production, on a hospital GMP production, or we can go to a CMO and take all processes over, make something easier, but it is not the case. But at the end, we want to have the product for the patient, for all patients in need available. And here we have, let's say, two ways. One is to focus on near patient and stage production, which is a strategy of some companies. Milton is one example. Or you have what the big pharmas are looking for, off-the-shelf delivery of products. So, with it in mind, we started several years ago together with Johan van Elderen. This is a UHA, let's say, consortium. This is a European Hospital Alliance, in which these universities around Europe are interacting and close connected, and within this European Hospital Alliance we built up the UCAT as a virtual European institute, again to transfer manufacturing technologies among us and to develop products together to advance clinical stage. And thanks to the European Commission, this, let's say, UCAT consortium get now the funding for CSA within the Horizon Europe. And I see it because it was a long way. It was not easy to achieve it. And we are leading this joint for ATMP. And I think to not only to say how good we are, but to convince you that we are able to set the right points also regarding networking, because networking is everybody speaking about, but in most cases it is not really a network, because everybody is claiming their own interest and looking only, let's say, to have the money for their own interest. But for us, for me, networking is more. It's really to build up infrastructure globally, at least in Europe, and focusing, of course, on different medical needs and aspects. And this is what we have established. We are leading, and here we are, luckily, I have to say, really luckily, partner in this kind of widening program. And I can promise that we do our best to help with our knowledge. This is very brief what our expertise are. Our expertise is related to enhanced targeting of cell therapies. It means redirecting of specificity. Homing aspects play an important role. Do the cells really home to the targets they have to go? And the lasting intratissue functionality, because mostly the cells come in an unfriendly environment, which means in inflamed area. It is even the case in cancer. So that you have at least a local, mostly also a systemic inflammation which target the cells you prepare outside. The second point is, of course, the viral vector gene editing or vector-free. We are focusing, as you know, you visit us already, more or less on the non-viral vector gene editing strategies. How you enrich the cells, this was a topic of Jürgen already. Yeah, and you can be sure that you never get a pure product. Yeah, this will not be, but you have to be as sure as possible. And this is also technology related. That your sorting strategy is really convincing you that you have the majority of the right cells you want to have in your product in place. Expansion strategy and of course single cell analysis, and Jürgen already mentioned the importance of preclinical models, and here I think organoids are already mentioned, and human organ on the chip is also one point. So this is more or less my last slide, because I think this is also a message towards the European Commissioner or Commission. For me it is not only an honor and a privilege to be here a partner, it is also one of the best dissemination strategies we can have, because in all EU consortia you have a huge part of dissemination. Yeah? So, but this is indeed dissemination, because we have to setting up GMP facility, we help in innovation, yeah? We provide contact with regulatory authorities, which is very helpful. You can make a lot of mistakes and turns with them. I think we have you more than we, experience with industry and VC, which is of importance with respect to the sustainability. Startups and training are already mentioned. So that's what I have to say shortly, more or less. Thank you.
Professor Reinke, thank you very much. So, kakšno vprašanje, any questions, comments?
I think everybody is tired.
That's also true, I guess. So we have our last. Yes. Professor Bonifacio from the Technical University of Dresden is joining us via teleconference.
Thank you very, very much. Sorry, just can you hear me, and can you hear me well?
Yes, we do.
Great. Thank you very, very much. I apologize that I'm not there. I'm actually on vacation. It's my only week, and I've not had a single day of vacation while I'm on vacation. But so it's fine. I think we're all used to that. And so a little bit about me while I share, if I'm allowed, grant access in the system. Allow WebEx to share. Okay. Good. Oh my God. Here we go. Sorry about this. Let's see. Okay. Later. Let me see. It won't let me open. Why is that? Because it's told me that I need to get out of WebEx in order to do that. I don't understand why. I do. I really can't open this, and I'm not sure what to do.
There is a chance, Professor Bonifacio, perhaps we can continue with Tanja Španić, We try to figure out what happened, and you present in the end. Is that an option for you?
I only have another 30 minutes, and then I can't present anymore.
Understand.
So I have 30 minutes.
Is it possible for you to explain via your power of storytelling? I understand it's not the same.
I can tell you about
the main points and your exactly. Thank you. Please do that. Thank you very much.
That's what I'll do. I'll look at my slides and tell you all about them. So just a little bit about me. I'm vzivo. You can tell that I am by my accent that I'm actually originally Australian. I'm not German, but I have an Italian background. And I actually was in Milan at the San Raffaele Institute, which I'm sure you all know very well, for 13 years or so, some time ago, and I was director of their pilot transplantation program. Why I have a strong interest in cell therapy, and of course partnered the Telethon Institute of Gene Therapy, so interest in gene editing. And I'm in Dresden. In Dresden, we are at the Center for Regenerative Therapies, and we in Dresden have a few years ago come together with our colleagues in Leipzig, and this is a venture that is in Germany is called a Cluster for Future. It is a venture which funds a joint partnership between academia and industry in order to develop industry in a region. And of course, the region is Saxony that we're trying to develop, and the area is cell and gene therapy. And Ulrike Kohl, who's at the Fraunhofer Institute for Cell Therapy, is my co-speaker in this venture. I think it's an important venture because it's very much dealing with some of the things that we have been talking, or you've been talking about today, and we will be doing over the next years, and that is to move things not just scientifically, but towards a therapy for patients, but also industry. And that's a real challenge, as you know, because scientists don't think that way, or at least the ones I know, so it's a real culture change for many people. As I said, it's a partnership between Dresden, Leipzig, and also Chemnitz, and a large number also of industry. I think I don't really want to go too much into the areas, except to say, obviously, areas of focus for us are the CAR-Ts, the CAR-NKs, but also other cell therapies, including macrophages. We have a program that is trying, like many people, look at universal cells and also modulating therapies. I did want to speak a little bit about the platforms that we have, or some of our core areas in Dresden, where we think we can contribute to this consortium based on where they've gone so far. One of them is the area of gene editing or genome editing, and for us it's not all CRISPR. We have Frank Buchholz as a major player in Dresden. Frank Buchholz is designer recombinases. That's what he's researched for more than 2 decades, and is really an expert in that, and has brought that to therapeutic levels where it's very predictable and precise. So it's It's very, very safe. It takes longer than the CRISPR, but it really can address almost any target. You can excise unwanted DNA, for example, HIV, which patients have in them, which he can remove, and you can insert into safe harbors wherever you decide. You can invert, for example, with factor VIII, and you can exchange any mutation. So this is a technology that is built around Frank Buchholz's core. He's also spun out companies, Provirix and more recently Seamless Therapeutics. And so it really is an example of taking something from the science right through to company and also therapy. The other area that is important for us, where I think we made some important contribution, has been in the what was originally called the UniCAR models, so modular therapy, where there is an adapter molecule that is the versatile part of it. It's controllable. Where the CAR T is only half of the matter, the adapter molecule is what activates it, targets it. And that is in clinical trials. Of course, there was a startup, Gemoab, which is now Avancell, sitting in Dresden. But these are a couple of our areas that we're happy we're sort of proud of and are looking forward to contribute. Through Saxicell, we've learned how to cooperate among competing places. That's actually never as easy as you like, but it has been a learning process. And we've also created a hub or a place of coming together, where we have tasks which we do together, which is to manage and communicate with the individual projects so that they are moving towards their goal, looking for venture capital, looking for that outreach wherever that's required, both at the industry side but also public and government side. So this is done, I think many places from the talks I've heard have this, and I think this would be a great share opportunity amongst us, because it is an important part of what we're all trying to do. We also have some platforms, we call them, clinical trials for the omics, which is the monitoring, and for systems, which is very much the automated processing of cell production. Some of the more novel areas that we have, I'm looking at my last slide at the moment, so one of the more novel areas is, of course, that our vision, like yours, go beyond CAR T cells, rare diseases and cancer, and we do look at new cells. We have Mikhail Sivica, who is with us and has renewable sources of macrophages for macrophage cell therapy, where these are gene-edited macrophages, and is taking that to a startup. We have Anke Fuchs, who works more on the immune dampening side of things with other cells, Tregs, for example, etc., and some other interesting ideas. So we're very, very happy to be part of this. Very happy to be with such excellent partners, and to be building something which we all know is important and has an enormous future for both patients and industry. So thanks very much. Sorry about the slides, but maybe nearly got you on time. Thank you.
Professor Bonifacio, thank you very much for the words. What I will remember from your speech is the need to learn how to cooperate. That's one of the important things. It seems that science and also progress in healthcare is a matter of complex systems, which mainly means that you actually don't control most of the system. So this refers back to the story of the ecosystems. I think this is the direction that is somehow visible and felt in the last years, not just today. And with that, thank you very much. And I would like to introduce now our last speaker. Actually, the closing remarks comes from a person I really don't need notes to introduce. We've actually been working together, I as a volunteer and her as the president of Europa Donna Slovenia and also Europa Donna Europe. We've been working together for about 5, perhaps 6 years, and it's a big privilege. I still learn from Tanya every week. We are mostly meeting on a weekly basis. Thank you very much for the opportunity to work with you, Tanya. I hope the guests here will also take something away from your closing remarks. Thank you very much.
Hello, everybody. Gregor, thank you for this nice introduction. I will conclude in English on behalf of our international guests. Thank you also, organizers, for inviting me and give me the opportunity to say something about patient perspective or expectation from this kind of centers and therapies. I earlier saw your disclosures, Jürgen, you showed. I always put them, but my biggest bias and disclosure is that I'm a patient myself. Exactly 15 years ago today, I was diagnosed with breast cancer. Now I know that that is really an early age. I was 26 at that time. So at that point, I just wished that I will live until I'll be 40 and that by that time I will throw a party. So I did. Last year, because of the COVID, I was 40 during the COVID, but last year my husband and me, we had a big party for our 40th birthday. It was actually a concert. Gregor was also invited. So celebrate life, live, survive. Do we want just to survive or do we want to live? My second disclosure is that I'm a scientist, or I used to be a scientist. I have a PhD in molecular neuroscience. Another Gregor was my professor during my PhD. And another Gregor, which happened to be a rector today, was my mentor. So I quit science. Did I really? No, I didn't. I still am a scientist. I still love science. I'm still part of many steering committees of research projects. But my profession now is a patient advocate, patient representative. Using all my knowledge that I got during my trainings to make this environment better. Maybe we can thank European Commission and EMA that they put it in their actual guidelines that there is really nothing about us without us. Earlier, Gregor also mentioned that at some point everybody is a patient, even if we're not aware either we are using a healthcare system. But today maybe you feel healthy, the next day you can get a diagnosis of cancer or any other rare disease. In that case, definitely research, development, and everything regarding the new technologies is very welcome. At the beginning, a few times it was already mentioned how patients are important and that we are in the center, but actually during these past years we learned that we actually don't want to be just in a center of the circle, but we want to be a part of it. Being involved in research from the very beginning, being involved in policy making from the very beginning, because at the end, you are the one who are receiving the treatment. And also development of the treatment, how it went from one size fit all to more tailored, personalized, individualized treatment, whatever you want. But this is nothing possible without science and research. Do we understand science really as a patient? No, we don't. But we are maybe in some part more emotional driven or passionate driven, like Spela, she was mentioned earlier several times. And this is good. But on the other hand, that's a huge jeopardize. We are too passionate. So we want the solutions very quickly. And we saw that bringing something from lab to the bedside takes more than decades. So we have knowledge, we have practice, but we have to bring this in the real life in hospitals and in treatments. Of course, our wish is that every single treatment, especially in rare diseases and in cancer, in neurology, pediatrics, should be tailored, personalized. Gene sequencing is something very common today. We know that some kits or some NGS takes us less than €100, $100, let's say, forget the whole genome sequence. So where are we today? Are we going to do more PET scans? Are we going to give people chemo if we need it or not? Are we going more to the tailored and precise treatment? Definitely this is the answer. And what we expect from centers like this, from cell and gene therapies, is that patients get their treatment at the right time, for the right diagnosis, and to survive, which makes her live, not just to survive. Especially cancer treatments and rare disease treatments are usually very heavy, long. My treatment was more than 10 years. So you can imagine that I live with several side effects, that I can live with them. You cannot see that I have more than 80% physical damage on my body, and I'm a disabled person. So, but this is something that you learn to live with, and that quality of life is something that you can survive and live this life. So that's all from my side, and I really wish this center to become successful and that we see the first patient get the treatment from this center in the next at least 10 years, and that this will become a reality. No pressure. You already said that you're under big pressure, but yeah, thank you.
I think that Tanya showed you another one of her talents, and not in the language of the boy, I mean of language, but she was dancing on the floor. So she used to be a dancer. She wanted to be a dancer. Now she's a patient advocate. She knows a lot about science. So thank you for accepting the suggestion. And of course, without diminishing the role of other patient organizations, and everybody in the triangle that was mentioned here. In this regard, please allow me to use what Dr. Fürst said earlier when we discussed. We need to drive, I hope that we are capable of directing the development of what we are doing with the acknowledgement of the differences that are enriching us, and also that not all things are to be just described in words. Some need to be danced, some need to be felt, some are there in spirit or in the goodwill. This applies a little bit to ethics, it applies to culture, it applies to who we are as a society and what is the meaning of what we are doing here. I am very convinced that the meaning and the biggest celebrations in life are not those that have a big, nice, fat check behind them. We are aware that in order to do this, we do need the support of the European Commission, the state, and everybody else. Thank you very much. This was a wonderful kickoff from my perspective, and I'm really grateful for having the opportunity to be your moderator today. Upam, da vas nisem za komentarji pravzaprav mogoče zapeljal v smeri, ki ne bi bila zaželena. Pa, kakor sem povedal, hvala, da ste tudi ostali, hvala, da bomo tudi v prihodnje lahko sodelovali. Mojca, would you like any final words?
I would like to thank you, the ones who stayed here, and see you next when we start building the house.
Hvala lepa, hvala.